Good evening, ladies and gentlemen. This is Dr. James Lyons-Weiler coming to you from the WWDNYK studios in Pittsburgh, Pennsylvania. Tonight on Unbreaking Science, we're going to be talking with a scientist and a very, very well-informed citizen who is right on top of the science, and it should be a fantastic discussion. But before we get into that, I want to point out the world is coming to an end. This New York Times has, in fact, published an article entitled, "It has come to this, ignore the CDC." Now, before you get excited about this, understand that the authors of this are Harold Varmus, the former director of the NIH, and Rajiv Shah, who is the director of the Rockefeller Foundation. What that might mean to you, I'll leave it up to you, but if you read the article, you see that what they're talking about is testing. They're talking about the CDC's decision to be forthright about the problems with testing. And I've been harping on the problems of testing since March, at a minimum, and specifically the concern over the false positives, the role of the false positives, which they only seem to want to talk about the false negatives so that they, can track more people down who might have it. Um, Sweden, for instance, adopted Germany's excellent, very accurate RT-PCR test that was actually validated by January 16th, and the world started downloading the primer sequences for the test and started producing the test locally. I don't have time tonight to go into how Germany validated it in January 16th without an actual sample of the virus, but let's just say that the Germans are quite brilliant and they actually figured out how to use synthetic nucleotides to create all the experimental conditions to test the sensitivity, the ability of the test to detect the coronavirus in a sample, the specificity, the ability of the test to not detect it when it's not there, and they went one step further. They demonstrated 100% pathogen specificity. Also, later on, you're going to hear, if it's published by peer review, my analysis of the cost of the false positive. You're going to see a figure like this in that publication. To get to our guests here soon, I don't have a lot of time to go into this, but let's just say I asked the question that no one in public health, no one in medicine, no one in the news, no one in government has asked, which is, at what point in time in an outbreak does it become ethical to switch from diagnostic-based testing to screening the entire population or to just doing arbitrary mass testing of any kind? And the reactionary activists might say it's never ethical to do that, but that's not how we answer questions that matter to society. We answer questions by finding out, in my analysis here, that it depends on the sensitivity of the test. So this result that you see here has specificity of 0.99. The test is assumed to have a specificity of 99%, which is much better than what Dr. Singhang Li found at the CDC, and much better than what even the CDC is now saying about their test. But let's assume it's a pretty good test, and you've got an outbreak, and initially you have very few people infected. What this chart gives us, and it was quite an extensive analysis, is the percentage of the population that has to be infected before switching from diagnostic-based testing over to random screening or arbitrary screening or just some other mass screening using a diagnostic assay, using a molecular assay as a diagnostic. And the short answer is that you basically have to be beyond the point of herd immunity in the percentage of people infected for coronavirus, no matter whether the cost of the false positive is higher or lower than the cost of the false negative. I think it's a pretty smart analysis. I wish I could do it justice here, but I just want to let you know, IPAC, the Institute for Pure and Applied Knowledge, we're asking the tough questions and actually getting some answers, just like my guest tonight, Dr. Theresa Deisher and Bernadette Pajer. Hi, guys. How are you?
Hi, good.
It looks like Theresa might have frozen.
Oh,
She'd have to call back, maybe. Anyway, Bernadette, how are you?
I'm doing very well. Thank you, Jack. I've got somebody texting me, wanted to know how they can tune into this program right now. Where are you streaming to?
Well, you can find us on YouTube, WWDNYK Studios on YouTube, and you can also find us
Okay.
at WWDNYK Studios on Facebook.
Okay.
That's where you can find it. And if you find it, please share it.
I'm texting now to let them know.
All right, cool. So let's start talking a little bit about why we were going to have Dr. Deisher on, and perhaps she, maybe her battery died or something, and she'll be here with us in a moment. So what's going
Okay.
on?
Well, yeah, I actually had the privilege of having Dr. Deisher on my program 2 weeks ago at Informed Life Radio, and she was bringing up some pretty important things that reminded me quite a bit about what you say about the problems with trying to invent a vaccine for a coronavirus, for something with a spike protein. And so I really wanted to hear her approach as a scientist and researcher and her angle on things from her experience, and you talking about what you know from molecular biology to see where you meet. And that's what I'm really looking forward to hearing.
Great. Okay, so Dr. Deisher, you're back. I don't know what happened, but here you are. Glad to have you. So let's talk about you a little bit. What's your background?
I'm a molecular and cellular physiologist, obtained my PhD from Stanford Medical School in 1990. And I went to Stanford University as an undergrad as well. My background after graduate student has been largely in the commercial industry, biotech and pharmaceutical companies, more recently with Amgen. And I've worked broadly in the field on cardiovascular disease, working on beta blockers for heart failure when I was in graduate school, and working on drugs for cancer, chemotherapy-induced cardiotoxicity at Amgen. I've worked on autoimmune programs, immunology. So I've been, I guess, blessed to be really broadly exposed, particularly the years I spent at Immunex, which was bought by Amgen. And I was at Genentech in the late 1980s as well, at the heyday of successful biotechnology. So
Great. Great. Well, you may know that I published a paper called on pathogenic priming, warning that all of the immunogenic epitopes in the SARS coronavirus 2 proteome that are immunogenic. That's not all of them, but all of them that are immunogenic, except one, have high regions of homology in the epitopes compared when you make the comparison to human proteins. And I found that a third of the proteins that show homology to human proteins actually might lead to autoimmunity against parts of our immune system, different proteins in different parts of our immune system. And so Bernadette thought it'd be a great idea for us to get together and have a conversation about what you're finding, and your focus being on a protein called the ICAM3 receptor. Is that right?
Well, we are very interested in understanding, how the virus enters the body, because the disease is distinct from the SARS back in 2002, 2003. So everyone has just assumed that it's using the same receptor, which is a receptor called ACE2 that the earlier SARS used. However, if it used that same receptor, then one would expect the same biology. Unfortunately, we all learned the hard way. It's a very different biology. And what it looks like early on is more like high altitude pulmonary edema, or what
Hmm.
is also called silent hypoxemia. And in that case, when you put those patients on respirators, you actually can drive them into acute respiratory distress syndrome. So the biology, the pathology is quite distinct, and the cells that are targeted, that are infected, are distinct. And so, we just took a look at what might be a novel receptor, and there's a receptor called ICAM3, which is limited to pulmonary bronchioles, which actually looks like it's where the SARS-CoV-2 enters, not through the alveolar space. And then on lymphocytes, monocytes, and neutrophils, which are largely the cells that are infected by this coronavirus,
So
the
let's
SARS-CoV-2.
for our viewers, let's back up to why it's important that we distinguish that it's a receptor. What does the ICAM3 molecule or protein itself do? It's a glycoprotein, as I understand it.
Well, it's the most heavily glycosylated protein, so glycoprotein in our body. We know that those types of proteins that have a lot of mannose groups on them are good receptors for viruses to bind to. We already know that. It's known to be involved in two cells binding together, so cell adhesion. And it's also involved in, interacting with other immune cells. So it's found on lymphocytes, and its natural ligands outside of that cell-cell binding area are proteins that are expressed on what are called dendritic cells. And dendritic cells activate other immune cells. So they're sort of like the, they can activate all different kinds of immune cells if they become activated. So those are its natural ligands. And the concern, so it's important that all of the receptors or the correct receptor is identified. So for instance, a lot of screening work to identify potential interactions has used cells that don't express ICAM3. So that's totally left out of all of the information out there that everybody is sharing and working off of. And if you're targeting the wrong receptor, your vaccine and your antiviral work could have some safety issues and perhaps not be efficacious, but have some significant safety issues as well.
Okay. So
can I ask a quick question?
Of course.
So how difficult is it to discover what you discovered? So I'm thinking lessons learned in COVID, moving forward, if we have a novel virus appear, shouldn't step one be for some scientists somewhere to look to see where it is, what the receptor is as quickly as possible so we don't base our further actions on assumptions? I mean, can we add that to our list of things we learned here? Is it difficult to discover this?
It could be difficult to discover it because they made assumptions based on how similar the spike protein is to the earlier spike, and the spike protein is how the virus enters cells. And I think they're about 70% similar, and so they assumed that the binding and this spike can bind to ACE. So they just stopped there, right? And I think lessons learned is don't stop at any assumptions and really try to get a handle on what the disease actually looks like. So early on, there were emergency room physicians in New York City who were, calling alarms. Hey, this is not acute respiratory distress syndrome. These guys look like they've been in an airplane at 35,000 feet without oxygen, but they didn't listen to them. And so the lesson learned is that we can't predict how viruses are going to act.
Okay, so there are there's some things that we know of blockers that block cellular entry for viruses or help aid in the blocking of cellular entry, including the spike protein in coronaviruses. And so what's the significance? What's the practical significance to translational science, Theresa, when if you see that there's another cellular entry, what are we to do about it?
Well, so what is also interesting in this, disease is that the patients who develop antibodies early at high levels, not only to the spike protein, but it turns out also to the N protein, those patients should not get the disease, yet they go on to develop the most severe disease. So it's not just the virus itself, but it's the, it's the antibody response. And so why would patients with the highest antibodies be the ones getting the sickest? Well, the antibodies must the spike must be similar, as you said, to other proteins in the body, and antibodies to the spike can bind to those other proteins. So if ICAM3 is a receptor for spike, antibodies that effectively block spike from binding to ICAM3 are also going to bind to what are called DC sign and L sign on dendritic cells. And that could really cause immune imbalance. It could be very dangerous. So it's important, to know the receptors so that we can think about safety. And that is a concern with the vaccines. That the vaccines to the spike protein, maybe even vaccines to the N protein, in some individuals, not everybody, but that they could trigger disease at the same time that they're efficacious, right? So if they're
Right.
not efficacious, then they shouldn't trigger disease. But if they are efficacious, they might also trigger disease. And really high levels of febrile responses have been observed in the early vaccine trials. And it's particularly concerning with the repeat dosing, because usually the fever goes down after the first dose. And instead, with these vaccines, a higher percentage of people became febrile.
That's a very,
So,
very big concern. So let me tell you, just so people know what we're talking about. I only looked at the one-dimensional sequence similarity. What Dr. Deisher is talking about is also the three-dimensional structure of the proteins, an analysis that my analysis touched on a little bit, because we did look at the immunogenicity. But many of the functions of the human adaptive immune system might be impacted via autoimmunity against the very proteins that we need to fight off not only coronavirus, but other respiratory viruses and other pathogens as well. My list included MHC class 1 and MHC class 2 antigen presentations, PD-1 signaling molecules, cross-presentation of soluble exogenous antigens, as well as the EG phagosome pathway proteins. I mean, it's a third of the ones that just show immunogenicity and sequence similarity. And I might be selling my study a little bit short, but how would we? Well, I've already reviewed 3 vaccine development studies as a result of my publication, and I sent them back to the drawing board. And I said, you guys have to remove these unsafe epitopes. But we're never going to know if we get all the unsafe epitopes, are we? So you're talking about ACE3 receptor, not ACE3 receptor, I'm sorry.
ICAM3.
Yes, the ICAM3 receptor. And I didn't find that. So mine's just an exploratory. Darshika Duke did another analysis. She found other ones that might be in the list as well. So the list keeps growing. I just read a headline that the states across the United States are being told to get ready to start distributing a vaccine at the end of October. They're no longer talking about November. So it keeps getting, the politicization issue is clearly here. The superhero, I brought the vaccine to market so that I'm saving the world. This mentality is a very dangerous one. I don't like to dwell on the politics, but in general, it is not a very common practice for any vaccine to study to screen for unsafe epitopes, right? Other than the 3 papers that are rejected until they remove the unsafe epitopes, I don't know of any where they said, you know what, this one's too immunogenic. The mindset has been very linear. It's been very simple-minded. The more immunogenic a protein is in a vaccine, the better. And you just told us that you get a fever on the first dose, and then on the booster dose, if you get a larger fever, that's atypical, and that's a bad sign. Things are going wrong. But these patients where we also see the adverse events with the most serious disease in COVID-19 infection might also be the ones that have the most serious health outcomes due to genetics to the vaccine, because their proteins are just a little bit more similar to these, and there's so many targets in the human genome. By the way, those papers, the editors of those 3 journals agreed with me, and 2 of the papers are now published without the unsafe epitopes. But they're not American vaccines. They're not the ones that are on the market now. These are other vaccines from overseas.
Right. Well, so, the vaccine trials right now are in healthy volunteers. And so, and in trials where they're not seeing fever, they're actually putting them on acetaminophen to prevent fever, which is concerning too. And so when you go out there into a general population, the sickest people are going to be the ones potentially adversely impacted by a vaccine. And I think, you know, we were caught unaware with this vaccine. And let's just say hindsight is going to be 2020 vision. But we have a lot of information now, and it's time to start reevaluating our response and what is actually the best response going forward now, and making changes, not only thinking about infectivity, thinking about mental health issues from what has been done, thinking about the impacts on our children and their psychological growth and emotional growth, thinking about the economics, thinking about the impact on our elderly, where dementia has gone up because they've been so isolated. And the CDC, and this is not really new news. This is just first publicly acknowledged. So first of all, the death rate this year is no higher than it has been any other year. And only about 10% of the people who died from just COVID-19. And everyone else had comorbidities. So will a vaccine go in and harm that same population with comorbidities and maybe do more harm than good because it's going to maybe be universally given, and so then we'll have higher deaths? Right. Do we need a vaccine, right, for a disease where only 10% actually died from the disease? And it's horrible.
What did you say? 10% died from the disease? Is that what you said?
All of the other people who died classified as COVID-19, there was an average of 2.6 additional serious comorbidities, and they probably would have died anyway this year.
Right. And so there's a couple of things you said that are worth unpacking. So the died with versus died from issue. In April, Dr. Birx made the announcement that, I'm going to have coffee too, all three of us. Cheers. We're having coffee together. That's awesome. Okay. So Dr. Birx made the announcement that they were going to start, if you test positive with a molecular test for COVID-19 and you died, they're going to say you died from that. It doesn't matter if you inhaled the virus 2 minutes before you died, or if you were, in one case in Florida, died from a motorcycle accident. They tested your cadaver and you happened to have an infection that had nothing to do, as far as we can tell, with the accident. The other thing that you said was, is it appropriate for us to even consider a vaccine? I'm trying to upload a study. I'm just going to read the title. It's by Peter McAuliffe at Harvey Reich, people that we know, Yale Community School of Public Health and Baylor University Medical, pathophysiological basis and rationale for early outpatient treatment of SARS-CoV-2. Okay, and what do they use? Hydroxychloroquine. This is a peer-reviewed study. It is published in the American Journal of Medicine. Month and a half ago, American Journal of Medicine published an opinion article that said that there should be a moratorium on the prescription of hydroxychloroquine. Just stop prescribing it, even if you're going to use it for malaria. Just no more prescriptions. And this is unprecedented to say, listen, we know what's best in every situation across the United States, all 50 states, all cities, all counties, every person. Nobody's going to get hydroxychloroquine. When all of the studies, the majority of the studies in the C19study.com website, over 50 studies show that it's not just the Detroit study at the Henry Ford hospital. It's study after study after study after study after study. There's many more studies that show that hydroxychloroquine, early use with zinc, early use prophylactically, not late use with appropriate doses, not lethal doses, like Dr. Meryl Nass came here on this show and said that that these were lethal doses that they used in the fraudulent studies. There's more science that show that hydroxychloroquine is effective against coronavirus than there are the so-called studies that show that vaccines don't cause autism by far. by far. So who's on the bully pulpit of science here? The public deserves to know, needs to know that the medical options include early outpatient treatment. If this is outpatient treatment, it should be over the counter. It should be just go pick it up at the drugstore. Not a problem. I'm going to share
Yeah,
in the just for
yeah,
the viewers, I'm going
I,
to share in the discussion. I'm going to share the link to that study that I read because I didn't bring it up on the screen.
so, one thing I think we want to remember, and that's why I really want to push everyone to just be calm and step back. Don't be wedded to your earlier ideas or opinions, right? Let's be data driven. Let's take a look at the data. But so many of the people in working with the people in the government through this, they are drinking from a fire hose, and they were working 7 days a week, 18 hours a day. I cannot tell you how hard they were working. And, someone may have seen the hydroxychloroquine studies where, it was very dangerous, and reacted to that without stepping back and looking at the whole picture. But, they might not have had the time. And so we need to just push on our government and say, okay, things have kind of calmed down. Let's readdress this. Let's take another look, and see if we can get this. See if we can get this right. I do see the CDC. I do see, other government organizations on a federal level becoming more circumspect, right? More data driven. And I just want to remind us, we cannot imagine how, I know how I was working, and they were working harder, and I didn't think I was going to survive it. So that's just how hard these people have worked. We need to say the same thing to all the governors. Okay, you didn't know, you thought you were doing the best thing, but here's the data. What you're doing is harming people, and there's no scientific basis for it. But I think they could readdress this.
Yeah, as far as trying to, I agree with you to a certain extent. I believe that there are a lot of fabulous scientists, researchers, people working at all levels who are earnestly doing their best with what they have. But I have been sending data and science and the effective working protocols as they are published to Governor Inslee, to Secretary of Health Weisman here in Washington State, to the Board of Health, to the Department of Health. And now we're at a state with Informed Choice Washington where we've actually petitioned them to form a committee to bring in practitioners who have effective working protocols, and let's sit down and have a powwow so that they can exchange ideas and that information can be given to the public so that as many lives as possible can be saved. They are resisting this with every fiber of their being. The Bill and Melinda Gates Foundation's global arm, it's infectious disease modeling, idma.org. They are providing the modeling, the data, and the step-by-step direction of what the state should do. They have been doing this early on, and our governor is not listening.
Where did Bill Gates get his medical degree?
I don't know, but they are not bringing up any treatments. Right. Well, and as of just a couple of days ago, with their latest report that they provided to the state, they said, well, things are looking better, but it's because everybody's masked up and staying home. So everybody, we still don't have a vaccine, so stay masked up and stay at home. No whisper that there are effective treatment protocols, and because they don't want that. And then we've got the whole McKinsey group that is in there too. There's now at least $5 million worth of no bid contracts given to the consulting firm McKinsey in Washington State, and ProPublica did a huge investigation. They found out at least in June they had more than $100 million worth of contracts with various states, and they're just like providing pretty data in a way, and shuffling things. And they're also funded by the Gates Foundation. And so there's a certain amount of, we have to, as grassroots, get the information out there, because there is this systemic block to it. But I agree that the world is populated with earnest scientists who are working their behinds off every day to try to get to the truth. So, Dr. Deisher, can you tell us, though, hydroxychloroquine, I think it should be out there and being used. And I think quercetin and acetylcysteine and vitamin C, there's great protocols there too. But you also have a product, your AVM0703, that has the potential, if we could just get it used for compassionate use, to put an end to things as well. Can you talk about that?
Well, let me ask the question. Let me reframe that question. It doesn't necessarily have to have an EUA for off-label, if it's already FDA approved. And I don't want to put Dr. Deisher in a situation of regulatory scrutiny, so we don't want to make any particular claims. But what is this product? I've not heard about it.
So it's not FDA approved. It's an experimental product.
Okay.
And we are supporting compassionate use with FDA approval. So they are approving patient-specific INDs, investigational and new drug studies, and they're really rapidly turning the approvals around. They've been fantastic. So we have a small molecule we were developing for cancer, and the way that it works is it mobilizes your own body's immune cells to be super activated. And the immune cells that it mobilizes by nature are programmed to kill cancer or any abnormal cells. So a virus infected cell is going to be targeted. And so, we did apply to Gates Foundation for funding, and they turned us down. So I guess that means our drug really works really well.
I think so.
Anyway, we have actually really great results from compassionate use, a really nice safety profile, and we're working with the FDA to move it forward. So, excited. We are about to get approval for a full clinical trial, which we would start enrolling in the fall. But in the interim, we are willing to provide the drug for compassionate use. And right now it's been mainly used in cancer, but, could be used for severe COVID patients not responding to any of the other drugs.
Beautiful, beautiful. So can healthcare physicians contact you to enroll patients in the investigational drug?
They can contact us if they want to be an official site in our clinical trial. If they have a patient who has immediate needs, they can contact us about compassionate use. And we have all of the documentation and templates. And the FDA is really responsive on how to submit. And we have an institutional review board who is reviewing everything pro bono, free of charge for the compassionate use patients. And we provide the drug and our support free of charge to the patients who need it.
Perfect, perfect. So we'll also post a telephone number in the thread for people to get a hold of you at AVM Biotech.
Yeah, to our website, www.avmbiotech.com.
Okay, I'll put that on your thing here. So you've heard of other treatments. Did you see Dr. Brownstein's protocol that was peer reviewed? The high dose vitamin C intravenous and so on. Are there any reasons why someone on another treatment might not take yours, or vice versa?
Well, we're really focused on those patients who haven't responded to anything else and have progressed to severe acute respiratory distress syndrome. So our target are the people who haven't responded to, vitamin C. I guess you can't get hydroxychloroquine anymore. Quercetin, low dose dexamethasone is having effect. Remdesivir is, has some effect. So for those patients who don't respond, then our drug is an option for them. And we have a really rapid onset of action. So
Fantastic. So there's the www.avmbiotech.com. Any final thoughts? I know you have to get on to another meeting, or any questions you guys want to discuss further.
Yeah, I think, for most people, I see people everywhere. Bernadette and I have talked about this, in panic. And fear destroys your immune system. So don't be afraid. Be healthy. Take vitamin A, C, D, and E, and I guess there's some other things, and prevent yourself from getting the infection. So we're healthy people, and healthy people should be out there breathing healthy, fresh air, not wearing masks, not living in fear, and taking care of our immune systems.
That's a
They're
great call to
they're
the public. It's a great call to the
and they're
public.
pushing everyone to take the flu vaccine. There are old studies that show that if you have a flu vaccine, you're 5 times more susceptible to coronavirus infections. We don't know about COVID-19,
Right.
but to coronavirus infections. So I think that's really dangerous to be pushing that.
Yeah, I mean, it's the most intuitive thing to say, listen, it's a respiratory virus. I want to protect you from another respiratory virus. But, with there being thimerosal in the flu vaccine, and the effect of thimerosal in inhibiting ERAP1, this is a protein that's responsible for folding proteins in our immune system. There's plenty of studies out there that show this viral interference or some effect of the flu vaccine making you more sick. I did a population-wide analysis using CDC's data a couple of years ago, and I found that if you get the flu vaccine, you're more likely to get an influenza diagnosis 2 years following. For 2 years, this is a long-lasting effect. Your earlier comment was a great call to the public, but I think it's time we turn to the medical community. They need to do their part too. And as much as I know thousands and thousands and thousands of American citizens through social media who can cite the studies on hydroxychloroquine, who know about the Brownstein protocol, right? Things are blowing up in terms of the public awareness. Well, it's the medical community's responsibility, like those doctors did that went to the steps of justice. Dr. Simone Gold, Dr. Emmanuel. Her middle name is Grace. The first name is her last name is Emmanuel. I forget the first name, but it's Stella. Dr. Stella Grace Emmanuel. Her complaint against her was just dismissed by the Texas Medical Board. So it's about time that we stop looking to citizens as the people who take on the full responsibility of getting the word out. The medical community better step up. The American public already distrusts doctors. They already distrust the CDC. And I'm looking at, the new normal as a world in which we don't have a CDC. I'm sure that our colleagues who are at the CDC that are frustrated by the fact that they might want to do something, but they have to follow protocol, are pretty annoyed that Dr. Fauci took control of this whole thing and took it upon himself to make medical decisions on behalf of all American citizens in an unfounded manner to bias the situation towards the need for a vaccine. It's right there. You can see it. It's not a conspiracy. It's 100% clear that the only motivation would be, and he actually said it, if he comes out like he did and he said, we don't want too many people to get this because then we won't need a vaccine, then that means that he doesn't want too many people to get immunity because you get herd immunity because then people won't need a vaccine. I mean, the man's not that smart. Why are we following this man who's not smart enough to even cover his own tail when it comes to misleading the public? So my point is, I think we need a distributed network of research studies sites, not the CDC. First of all, it's strategically stupid to place all of our bio response, our bio threat response capacity in one geographic location. The CDC should not exist. There should be 80 different sites that answer to the Senate for their funding, not peer review. And what they do is they keep their tendrils out and their radar up for bio threats. And every year they make a new report to every Senate committee. Now that Senate committee then makes a report. All of those Senate committees make a report to the Senate Intelligence Committee. Those Senate Intelligence Committee members then make a report and they inform the President of the United States on what's going to happen. because the president of the United States is not a medical. There should never be a Fauci in the future where one person speaks for everybody. And what I just described to you is called a neural network. It's actually the structure of a neural network, and we would design the information flow in a manner in which it could allow the emergence of intelligence, no matter what the biothreat is. Okay, so I'm totally on fire about how this next future, who builds the next future? Are we going to just sit back passively? Are we going to allow them to say, oh, well, CDC didn't get this one right? I mean, hundreds of thousands of businesses have been shut down, many of them permanently. 50% of them are never going to open up again. And all of that, all of the suicides you're talking about, all of the depression and all of the disruption of schooling and all of everything, where that burden is the cost of the false positive. And that's why if this paper is published, it'll come out in a very prestigious journal. The journal publisher and editors have already, they see the writing on the wall of that analysis I just showed you, because I'm going to, if it's published, if it survives peer review, I'll invite other analytics to come in and look at the cost of the false positives from a 360-degree. What we do in America is we tend to externalize costs. The corporations tend to say, this is my bottom line, and we're going to externalize the cost. And what we've done here is we've said, nobody owns the cost of the false positive. It's not my responsibility if I put it out there in terms of a bad test. Oh, well, it's a bad test. I'm trying my best. That's not going to fly. The CDC owns the lost jobs, in my view, because they could have, should have. I know dozens and dozens of scientists who were ahead of the curve in terms of CDC on this. And what I hear is from CDC, yes, they're softening their stance, they're wavering, they're going back. Maybe we shouldn't test so much. My God, every time a kid tests for a false positive test for coronavirus, they're going to shut down the entire school. They're going to close a college. They're going to shut down a business. These are like little bombs going off. A terrorist organization from overseas could never have this effect on the destruction of our society and what we're trying to accomplish here in the United States. It's really, really beyond the pale.
Yeah. So a couple things. The CDC didn't shut down businesses. Governors did. And the governors have to take responsibility for what they did, shutting down the economy. I think, the CDC is there because the older, more diverse models where everybody reported into, senators who aren't trained to make any decisions didn't work. Not that the CDC doesn't need shape up or whatever. And Dr. Fauci isn't with the CDC anyway. Dr. Fauci is with the NIAID at the NIH. I think, there was so much panic with this virus early on that we got to give people the benefit of the doubt. And Dr. Fauci has actually backpedaled and even said he really doesn't think there's going to be a safe vaccine in the near future. And maybe we won't even need one, right? But the death rate of this vaccine was going up at rates astronomical compared to any other global pandemic we've ever had. And they reacted to that, and people overreacted, and there was so much passion and emotions. I know the meetings I sat in, and I just have to say I have to give those people the benefit of the doubt, and that they've calmed down now.
I appreciate that. I had my finger on the pulse from January on this, and I tracked. We knew by April, certainly by March, no later than April, that the death rate of this virus outside of the Wuhan district in China was lower than the death rate within Wuhan. We also know that the actual infection case fatality rate, we knew this, I think, by the end of April or May. It was pretty well understood that the death rate was nowhere near the 20% and nowhere near the 3%, depending on what your illness is. Then we learned.
We knew that. That's very true. But just think about this. As scientists, we knew that. We're looking at the data. We're staying calm, right? But we're not on the front lines. People always tell me as a scientist, you got to step back and you got to learn to speak English. and like our decision makers making decisions, some of them, they only have the bandwidth to absorb so much. They don't understand the science, and maybe they saw that curve and they cannot get over that, right? So they let their states down, and maybe it was more sinister than that. I'm not going to judge, but they let their states down. But I think that there's circumstances we just all have to calm down so that we can do the right thing going forward. Healthy people should not be quarantined. That's house arrest. Sick people should stay home. If people are afraid, go ahead and self-quarantine. But there's got to be a point where they do it on somebody else's dollar, right? Because nobody wants to work. They don't want to leave their homes. And all the taxpayers will take care of this for a disease that you're right. It didn't really kill very many people. A lot of dead people, no more than any other year, and most of the deaths because they were medically mismanaged, right?
And most of the deaths because they were old. The geographic location, there were 40% of the deaths in 4 states, the same states that Fauci said were doing it right. I mean, this is ridiculous. I know the NIAID. You know me. I served on NIH grant review panels. I understand the structure of that particular arm of the government. The point is, these regulatory agencies, the state, Fauci became the spokesperson for the CDC, effective the de facto, when the CDC shipped out the faulty test. The CDC shipped out a faulty test instead of adopting this test from Germany. CDC then, according to Chris Meekins, lied to Secretary Azar over the readiness and availability, or the readiness and accuracy of the tests. They shipped them out. People tested positive who weren't positive, negative who weren't negative. Those people, this is why it spread. We lost our opportunity to do contact tracing early on. This is all on CDC. Who came forward? Fauci came forward. He did a whole media tour. He was the apologist for the CDC. His quotes are, "There's no bad guys here. It's just something that happened. We shouldn't really look at it too closely right now. Now's not the time to troubleshoot this." Baloney. The time to troubleshoot it was right then. There should have been a Senate inquiry on this, specifically with, okay, your test doesn't work. What are we going to do? The best that they come up with was let everybody make their own tests. UPMC hospital, the major hospital here in Pennsylvania and worldwide, actually said, screw you to the CDC, screw you to the FDA. We're going to make our own test and start using it tomorrow. We don't care what you say. All right, before the EUA.
Well,
so,
the tests, there are other things here definitely going on. It will come out, but a virus is contagious. You just spit. You never stick a swab up into someone's olfactory mucosa where their most valuable stem cells in their body reside. So I've always had a problem with this test.
Me too. I early on saw the science that showed if you do a PCR test for coronavirus and you do the stick in the back of the that it's less accurate than a sputum test. Well, it only makes sense. You're going to pick up everything that a person has with a sputum test. So I don't know what who makes up these policies.
Well, most countries now are using spit tests.
That's good. I can calm down a little over the spit test, but they're still inaccurate. Did you see Dr. Singhan Lee's? What happened with Singhan Lee? We did a whole episode on it. We've got 2 minutes. I'm not going to recap the whole thing. But my God, the test reference samples sent to him by CDC were testing positive when they had no virus and testing negative when they did. So if that's the way CDC cannot validate tests if they can't make an accurate test. And I'm sorry, these people might be lucky enough to get hired at one of my 80 research nodes. We'll have to get a resume from them and see if they were making good decisions or not. Theresa, I'm going to give you the Bernadette first, and then Theresa, let's close out. Go ahead.
Well, I just have to say that I've always said that public health, lowercase, the number one enemy to lowercase public health is uppercase public health, because when government becomes in charge of anything and has billion-dollar budgets, and especially billion-dollar marketing budgets, you've created a monster with tentacles that reach into everybody's household. And putting a stop to that machinery is very difficult. And right now we're learning that. So something that they decided to do 3 months ago is still being flashed on overhead signs that, the marketing campaigns went out. It's hard to slow down this monster. We have to figure out a better way to be more reactive, in the moment, change policy. And the other thing is we can't ignore the corporate capture of public health. The Bill and Melinda Gates Foundation is running COVID response at the state level in every single state. So,
Right. So that network that I described to you, it's specifically designed to be highly adaptively flexible. The 80 nodes don't talk to each other. They can do peer-reviewed stuff. They can publish if they want, but their job is once a year to put together a bio threats report to the Senate committees, all Senate committees, so all interests of society get heard. All right. So that's an adaptively flexible thing. You get group thought, you get perverse incentives that we've talked about on Unbreaking Science. Go to unbreakingscience.com for three boot camp episodes, and you'll learn how they're doing it wrong across observational studies. Thank you, Bernadette. It was a pleasure to speak with you. Theresa, what's your closeout?
I have to drop off because I have a 5 p.m. meeting, so I apologize. Yeah, I agree with Bernadette, and anytime you give away a right, it's a problem. People think they're getting something from it, but oftentimes not. I would just say to people, don't be afraid and take your vitamins, exercise, eat healthfully, and it's unlikely that you'll even get it. And 99.95% of you will survive it. Anyway, we'll get into that much later, but just be healthy.
Yeah,
and
cool.
Don't be afraid.
Thank you. I like your message to stay calm, be healthy. We're going to find out that 0.28 infection case fatality rate is a way over exaggeration because so many of the cases simply tested positive for a molecular test. We don't do CT scans on every person every year for cancer because you kill so many people with infections from biopsy. To do a molecular test every day on so many people, you're finding all of these people that happen to have test positive. Maybe even if it's a perfect test, they're not sick. And so Theresa, Dr. Theresa Deisher, thank you. Bernadette, thank you. And rock on, guys.
Thank
Stay
you,
calm,
Jack.
Stay informed. I'm going to go have another cup of coffee and talk to you guys later.
Okay.